Hi, folks. Welcome to Naturally Speaking with Dr. Earyn McGee. And we're going to get started a few minutes past 6, just to give people some time to trickle in after work or after eating their dinner. So make sure you've got some water or some tea, get comfy, and we'll get started momentarily. Hi, folks, you're going to hear me say this a few times, but we'll get started shortly past the hour to give people enough time to trickle in after work, after eating their dinner. So now is the perfect time for you to grab a glass of water or a cup of tea to get comfortable, and we'll get started maybe 6:03, 6:04. Thanks for joining us. If you're just joining us, this is Naturally Speaking with Dr. Erin McGee. We're waiting for a few minutes past the hour to let people trickle in after work. So grab a glass of water, get comfortable, and we'll get started around 6:03, 6:04. Thanks for joining Naturally Speaking with Dr. Erin McGee. We are going to get started in 1 minute. We're giving people some time to log on. So get comfortable. We'll get started very soon. All right, everyone, we're going to go ahead and get started now. Thanks for joining Naturally Speaking. Tonight's speaker is Dr. Earyn McGee, herpetologist, ecologist, and science communicator. I'm your host, Sam Wins, conservation biologist by trade, also a science communicator, and I work at Cabrillo National Monument. And before we get started with Earyn's talk, we're going to do a little housekeeping items. So let's go ahead and move this slide forward. So this isn't a webinar format. You're not going to be able to see other participants, only the hosts, like me and Earyn. And to ask questions, please use the Q&A function. You can find that either at the top of your screen or at the bottom, depending on what type of computer you have and what version of Zoom you've got. And you can ask questions anytime with the Q&A function. They're going to be queued up. Earyn will pause periodically to answer questions that you might have. So make sure when you think of it to load them up in that Q&A. And then at the very end, we'll probably have a more robust Q&A at the end of Earyn's talk if we have time. Earyn might also ask you questions, right? This is a conversation that we're having. So make sure that you use the chat function at the bottom of the screen to answer Earyn's questions. Please keep your mic muted at all times. It can be disruptive if there's a bunch of people talking all at once. And captions are available by clicking the closed caption icon either at the bottom or at the top of your Zoom screen. All right, and without further ado, let me introduce you to tonight's speaker. So I am so pleased to have Earyn join us tonight. This is Dr. Earyn McGee. She's a herpetologist, an ecologist, and an amazing science communicator. She's an award-winning scientist and an award-winning science communicator, an If-Then ambassador. He was in Forbes 30 Under 30 lister for her amazing game, Find That Lizard. If you don't play, I highly recommend you check it out after this. And has been featured in places like Marie Claire and documentaries. So I just can't say enough about Earyn tonight and sing her praises. But enough for me, right? You're not here for me. You're here for Earyn. So without further ado, I'm going to turn it over to you, to Earyn. You are muted right now, by the way.
Thank you. All right, thank you so much for that introduction. And then I'm going to try to share my screen here. All right, are you seeing the presentation? Oh, wait, this is the wrong one. Sorry. Click the wrong thing. Go to
I
the
told y'all she does a lot of guest speaking, a lot of speeches, a lot of
Yeah.
guest speaking.
Me thinking I was like, oh, I'm extra prepared. Mm,
All right. Now's the perfect time for y'all if you have any preliminary questions for Earyn. What does it take to be a herpetologist? What is a herpetologist? Where does the term herpetology come from? Which is one of my favorite facts. Now's a great time to start queuing those up.
you- All right, there we go. Why not? Hmm, Sorry for the technical difficulties.
No problem.
Okay, let's see if that works. All right, now that should have the right one.
Okay. Lizards at Cabrillo and Science Outreach. That looks like it's the
Yes,
right one.
that belongs to you guys. All right. Well, thank you for that wonderful introduction. Sorry for the uh, not having the right presentation up. But I'm excited to be here today, and I'm so grateful for the opportunity. Talks like these are really fun, especially when I have the opportunity to do some research on some lizards that I myself did not research and get to talk to you guys about those. So, let's see, there we go. As Sam was saying, I run a game called hashtag Find That Lizard, and it's essentially like Where's Waldo, but with lizards. And so I go outside into not always into the wilderness, because sometimes we have our urban lizards. So I could just, sometimes go up the street to the park and see some lizards, and then I'll take a picture of the lizard and it's camouflaged in its natural environment, and then I'll post it and then people have to find it. And so I started this game on Twitter back in 2018 when Twitter was still Twitter and it wasn't X. And it was just kind of a way for me to bring people out into the field with me. I've been really lucky to have a lot of really great mentors and people who tell me about the unspoken rules when it comes to going through academia and job searching and being a scientist and things like that. And so I wanted to give back to other people in a way. And I was trying to figure out how I can do that while I was a grad student without taking away from my graduate studies and with having very little funding to do outreach. And very little knowledge to do outreach either. So I've definitely have grown a lot since I've started this or well, it's going to be a program. We're gonna call it a program. It wasn't a program before, but we're gonna make it into a program. And it's been a really great journey. And so essentially, I was out in the field one day and I saw this lizard and I needed to catch it because it was in an area where I didn't have a lot of um data points, and so each one was really important, and so this one would have been a recapture. And so every time I catch the lizards, I give them a paint mark on their back so that way I can identify them from afar. So I'm like, all right, it's time to catch this one. Start to chase her because lizards are actually really smart. People always talk about like lizard brain, but lizards recognize, I'm not sure if they recognize me as a person or if they recognize my lizard lasso, which I used to catch them, but they recognize something and they're like, never again. And so this one took off running. And so I'm running behind this lizard, trying to catch it. And it gets to a point where I'm like, I've lost it. I don't know where it is. And so I kind of scan the area one last time and I look over my shoulder and there's a lizard. And remember how I mentioned those paint marks? It had a bright orange 5 on its back, and that's what broke up its camouflage. And so I took a picture of that and then I posted it to Twitter. And I got a little bit of engagement. I got like 2 likes and a comment. And for whatever reason, the hubris of my brain was like, oh, we could totally turn this into something. Let's just keep doing this and do it regularly. And that's essentially how Find That Lizard was born. And so what I like to do is start presentations off with a Find That Lizard challenge. So let me know if you can hashtag Find That Lizard to make it a little bit easier for you. I gave you a couple of suggestions on where the lizard might be. So go ahead and put it in the chat. Where you think the lizard might be. And Sam, if you could tell me or tell us what everybody is thinking of where the lizard might be, that would be great. And we'll give you about 30 or so seconds to do that.
All right, friends. If you think it's in spot A, you'll just type A into the chat. If you think it's in spot B, type B, and C, type in C. Go. You've got about 30 seconds from now. They're thinking real hard, Erin, because there's no guesses so far.
No guesses?
I know there will be.
All right.
All right, friends. We're gonna do a 15 countdown. Ready? Like 15, 14, 13, 12, 11, 10. Just make a guess if you can't figure it out. 9, 8, 7, 6, 5, four, one. Thanks, Eve. Yeah, the chat is disabled, but you can put it in the Q&A for now.
Ah,
I
yeah.
will fix that chat disablement. Sorry, that came on apparently automatically.
Well, we'll just assume that people already had their answers. And
I
so,
see there are some answers in the Q&A. So there's
okay.
one, let's see, um, two for C.
Hmm.
So you got four for C and one for A.
All right. Well,
That's
if you
that on
guessed
Q&A.
C, you were correct. A was a little tricky because there was like a little piece of branch that kind of looked lizard-like, and you know what? Don't even feel bad because it gets me like that every time where I will look at something. I'm like, is that a lizard? No, it's just a stick that looks like a lizard. Is that a lizard? No, that's a rock. You know, that happens to me all the time. You just don't know it because social media is not real and I only post the stuff that I actually find. You don't see all the background where I'm walking around for 30 minutes asking myself if things are lizards until I actually find one. But it's a really fun game, and I've had so much fun doing it, and I've gotten a lot of positive feedback from people, which is really fantastic and keeps me motivated to keep doing it. And as I mentioned, I am trying to turn this into like more of a program and things like that. So, uh, this year, I actually hosted a scholarship and I was able to give out almost $3,000. And this was a scholarship for women and gender non-conforming people interested in herpetology because as far as I know, there aren't any scholarships, especially for us. And um, I specifically wanted to do something like that, something like this, because herpetology is a predominantly white and male field, and it is really important to increase diversity, not only because of the material ways that it helps different industries, not just herpetology, of like bringing in new ideas and coming up with new solutions and things like that, but also just because I think that this is fun. I do this because I love it. And I think that other people should have the opportunity to do the things that they love as well and should not be excluded from spaces just because of their race or gender or sexuality or ability or disability, anything like that. I think everybody should have an opportunity to do these kinds of things. So it was really amazing to host this scholarship. I got a lot of applications, and for the future, I'm really hoping to turn this into a nonprofit so that way I can go and solicit more scholarship money to give out more awards because I wasn't able to fund everybody this past year, but the goal is to one day be able to award scholarships to everybody who asks for them. So I'm really excited about that. So, getting into this presentation, we're going to talk about a lot of things. I'm going to give you some background about me so you can kind of understand who I am and my motivations for the work that I do. We're going to talk about some of my graduate research. So, I was looking at riparian lizards and their diets in the southwestern United States, specifically southeastern Arizona. We'll talk very briefly about the Doris Duke Conservation Scholars Program. Then we'll get into lizards at Cabrillo, and then a little bit about my outreach and career. So, who am I? I'm a herpetologist, science communicator, informal educator, advocate, community member, and creator of hashtag Find That Lizard. And I'm really influenced by a general curiosity about nature, my experiences as a Black woman in a predominantly white field, a desire to break down barriers, and just wanting to have a overall positive impact on the world. So I am not new to this. I am true to this. From as long as I can remember, I have always wanted to work with animals. So much so that when I was a kid, and so I was born in Atlanta and raised there for a little bit, and then we moved around, but I've settled in LA. But when I was a kid, my parents sent me to Zoo Atlanta, which is a summer camp that they have out there, which was really it was instrumental for me in that it was great that my parents were really supportive and let me have that kind of experience. to just keep that passion going, keep the flame hot, if you know what I mean. And so there I was touching a snake. I didn't know that I was going to get into herps when I was a kid. I honestly thought it was going to be more of like horses or like wolves or elephants. Like my parents are like, you can either be a zookeeper or you can be a veterinarian, but you really have to be a vet because zookeepers don't make enough money. And when you turn 18 and leave this house, we don't want you to come back. You need to be able to pay your own bills. And so I was like, okay, I'm gonna be a vet. I'm gonna be a wildlife vet because I want to work with, wild animals. But then I ended up going to Howard University, where I met this guy on my left-hand side here, Dr. George Middendorf, who was, or who is, he's not a professor anymore, but he's still alive. So he is a herpetologist and really accomplished. And essentially like meeting him changed my life. And part of that was because I was just telling people that I want to be someone who works with animals. And so an upperclassman mentor heard that and he was like, oh, then you should come meet this guy. And so like telling people about what you want to do is like so important. So like never be ashamed, even if you think that it's like just totally far-fetched and out there. Like you never know what kind of opportunities will come just because you're vocal about your interests. And so I met George and I started working in his lab doing data input. And then one of my friends, who I also told that I was interested in working with animals, told me about this program called the Environmental Biology Scholars. And he had to tell me about it because my GPA was not where it needed to be. So I didn't get the initial email. So when I found out about the program, I actually had to go to the person who was over it, Dr. Mary McKenna, and essentially advocate for myself and be like, hey, you won't regret this, I promise. If you let me in, I really, really want this, and I really want to be in this program. And so, uh, it took me a while to get my GPA up where it needed to be, but I think that she's probably pretty proud of me at this point. So that was really great. Um, and then skipping a couple more years, I went to grad school. And so I met Dr. Michael Bogan, who ended up being my grad advisor because he knew George from the Southwestern Research Station, where I did my undergraduate research. And George has been there, going there for 40 plus years, doing his research. And George and Michael had met when Michael was starting his lab at the University of Arizona, where I went to grad school. He was like, hey, do you have any undergraduate students who are interested in grad school? And George was like, yeah, I have this one right here. And so me and Michael met, we hit it off, and the rest is history. He's been such a great supporter of mine, and so if anybody here is looking to go to grad school, I would highly suggest finding a supporter, finding an academic an advisor who is really going to have your back because that is going to make or break your experience. Like I hear a lot of grad students talking about like how they had a terrible time in grad school, but I honestly had a really exceptional time. If grad school paid better money, I would still be a grad student because I absolutely loved it and I was very well supported in just being able to explore different concepts in ecology and then also pursuing like some of the social sciences type things. So now we're finally getting into the lizards. And so I'm only going to talk about this first chapter of my dissertation, where I was interested in if lizards were eating emerging aquatic insects from stream systems, with the idea that because of human-driven drought and stream drying, they could potentially be losing that food source. So I wanted to know, are perennial streams, so streams that have access to water year-round, providing aquatic subsidies, aquatic insects to riparian lizards, will lizard abundances be greater in perennial resource? Um, will individuals within the species grow larger and faster along perennial streams as compared to ephemeral streams? So ephemeral just means that it does not have access to water year round. So, There's been a lot of studies on how terrestrial subsidies impact aquatic systems. So stuff like leaf litter, um, uh, any kind of like animals falling in to be fish food and things like that, how those impact streams. But the opposite hadn't been studied as much. And a large review paper, so a paper that looked at data from a whole lot of other papers, um, found that the loss of aquatic prey could negatively impact terrestrial species, including lizards. And lizards are important to our ecosystems as predators, as prey, for nutrient cycling. And then also, it's easy to look at lizard populations and try to extrapolate or make statements about the ecosystem because you have a lot of lizards, they're relatively easy to catch. Whereas if you're trying to understand big questions from, say, like mountain lions or things like that, they're generally harder to study than, say, a lizard. So I did my research in the western US and southeastern Arizona on the border of Mexico, New Mexico, and Arizona in the Chiricahua Mountains, and it's part of the Sky Islands. So Sky Islands are essentially mountain ranges that are separated by large swaths of desert. And so the mountain is the island in the desert. And so at one point in time, it wasn't desert, and so the different species were able to move across the land, go down from the mountain onto a lower area, lower elevation area, move across and go up to another mountain if they wanted to. But now our higher elevation species, which are more adapted to colder climates, can't make that trek down the mountain and across the lower area, the lower elevation area. So now we kind of see speciation, so different species becoming more different than they are alike and things like that. And so that makes that area really great for biodiversity. And there's a lot of really cool lizards that are only found in that area and when it comes to being in the US. So it was a really awesome time to be out there, and it's really beautiful. So I was interested in three different species: Scalopus yervi, yarrow spiny lizard, Scalopus virgatus, the striped plateau lizard, and Urosaurus ornatus, the ornate tree lizard. And I picked these three lizards because they are the most abundant in the area, and they were also the species that I studied as an undergraduate student. And they are relatively easy to capture, especially because they like to go out and they like to kind of like bask on their rocks or their trees, and they're not as flighty as, say, something like a whiptail lizard. So, uh, and then in my study sites, I had perennial, so again, areas that had access to water year round, and ephemeral stream reaches, and they were 100 meters each, and I had, depending on the year, I had either 3 to 4 different sites, and those were each about 100 meters each. And so what I would do is I kind of divide the study site into like three. And so like I had a line that went down, an imaginary line that went down the middle, and I would walk that imaginary line and I would look for lizards on my right and I would look for lizards on my left. And then I would have another a line a bit out on the right hand side, and I would do the same thing, look for lizards on my right, look for lizards on my left, and then again, on the left-hand side, I had a line that I would walk, look for lizards on the right, look for lizards on the left, and I would walk up 100 meters, and then I would walk back the 100 meters, and then I would uh, be able to capture the lizards as I saw them. So for lizard capture, I used my lizard lasso, and once they were captured, I would take their mass, their sex, their snout vent length. So the vent is, or cloaca, is where the the end of the body and the beginning of the tail is where that spot is. And then I would also like do tail length, look to see if they had any like parasites and things like that? Did they have regrown tail? I was interested in how many lizards were in each area, so lizard abundances. And then to look at their diets, I was also collecting insects with emergence traps. And so essentially that's what that thing is on the bottom photo. So I took PVC pipe and I created a pyramid shape with it, and then I draped this mesh netting over it, and then I put cool foam floaties on the bottom of it so that way it wouldn't sink. And then at the top of the pyramid was a bottle with a hole cut out, and then it had alcohol in it. So as our aquatic insects moved from their larval and water stage into their adult terrestrial stage, they would climb up the side of the PVC pipe to the top to emerge, and then the fumes from the alcohol would make them fall into the bottle. And so then I would just go and collect the bottle so that way I can know what aquatic insects were in those stream systems. And for those of you who don't know, well, before I say that, the reason why I was collecting the insects was for stable isotope analysis. And so that essentially was letting me see what the lizards were eating. So from the lizards, a form of a more less invasive form of permanent marking is taking toes. And so you will take like a piece of their toe right after the nail. And so that still has tissue on it. And so a lot of diet studies normally either euthanize the lizard so they can look at their gut contents, or they do gut pumping, which is shoving water down their throats to make them vomit. And I was like, well, if we're taking toes for permanent marking, anyway, then how can I use stable isotope analysis to look at their diet so that way I can be as little invasive as possible? So then I want to go backtrack a little bit into their capture. And so I've been mentioning my lizard lasso. And so this is the lasso. It is a telescopic fishing rod that I got from some kind of uh, fish outdoor store in Arizona when I lived there. I don't remember which one it was. Um, and then it has a line at the end. So you can use a lot of things. You can use fishing line, dental floss. Um, this is braided surgical silk that I got as a gift from my undergraduate advisor because it is very hard to come by because now most surgical silk is actually already attached to suture needles. And so this is really nice because it holds its shape really well. And it doesn't, and it moves very like smoothly along itself. So essentially, you're fishing for lizards because when a lizard is coming along, or when you're coming along and you see the lizard there, what you want to do is to get the loop here over the lizard's head and then pull up. Oh, I got its foot a little bit there. And that's how you know it's hard. It's not even a real lizard and a struggle. And so then you pull up, and so because of the lizard's weight, it collapses the lasso, but the lizard is actually it's heavy enough to collapse the lasso, but light enough where this actually does not hurt them. And so historically, this method was actually called noosing. And for me, as a young Black woman in the United States, in the middle of nowhere, with no other Black people in sight, that made me feel very uncomfortable. And then when I would tell people about these methods, and doing like outreach events and stuff like that, people will often be like, "Oh, are you hurting the lizards? Are you killing the lizards?" And that kind of like really stopped the outreach portion of things pretty abruptly. And because I had to really like tell them like, no, we're not hurting them. And then it's like rebuilding that trust with them as a scientist of this is a necessary thing and it's also not hurting them and that type of thing. And I'm already doing clippings. I'm not really telling them about that bit of it, but it's just like, if they ask, I'll mention it, but it's just kind of like we're trying to let them trying to have them understand what it is that we're doing and why we're doing it. So it's better to use language that makes sense the first time. So because I had already been on social media, I was just kind of like, okay, well, this makes me feel uncomfortable. This makes other people feel uncomfortable. And we also say we want to be a more inclusive career. So how do we feel about changing this language? And so I'm not the first person to use lasso or to come up with it, but that kind of seemed like the consensus on Twitter, at least, of what people were comfortable with using. And so that kind of instigated a language shift to be more inclusive and to better communicate with the public about what it is that we were doing. And I'll stop there if there are any questions. Otherwise, I can continue. Sam, if you could let me know if there's questions. I just have presenter view going on here, so it's hard to see the chat. You're muted.
I clicked it, but it didn't work. Okay. Yeah, I see no open questions at this time. So I think we can move forward, but remember, folks, to add your questions to the Q&A, and Erin will pause periodically. Like, I'm surprised that no one had a question about what stable isotope analysis is.
Yeah,
So if you are one of those people who are wondering, Erin can talk about that. Please answer your question in the Q&A. Go
absolutely.
for it, Erin.
Cool. All right, so then moving on. So for the lizard abundances, we didn't really see any significant difference between lizards at our perennial or ephemeral sites. And before I did have three species that I was studying, but I didn't capture enough to include the third species, the tree lizard, into the analysis. So that's why they're not mentioned here. And then we did have like one site where we had just all the lizards were there that year for whatever reason. And so that's what that little outlier is there. And but we did see um a slight significant difference for Scloppers yarrow spiny lizard when we were looking at mass between perennial versus ephemeral sites, but that pattern did not continue with Scloppers virgatus. So that tells us that maybe there's something there. I would love to continue this research because I think that if I had more time at each site and less sites potentially, that I would have captured more lizards to have more concrete data, but it was still interesting to see that there was some significant difference somewhere in there. And so here's the stable isotope data. I'm not really going to go through all of this, but there was no real significant difference there. And so essentially we had like a bunch of different aquatic insects and terrestrial insects that we were looking at. So um, ants, beetles, wasps. We had aquatic flies, mayflies, which are another aquatic insects, caddisflies, aquatic moths, things like that. So we were capturing we think that we captured a pretty broad types of food items that they might be eating. So overall, we didn't see a significant difference in lizard abundances between perennial and ephemeral sites. Scloppers scierivi were larger at perennial reaches than ephemeral ones. However, this pattern didn't hold for Scloppers virgatus. And then the lizards were mostly eating the same things at both the perennial and ephemeral reaches. And we think that that just has to do with the fact that one, our lizards are sit and wait predators, so it's whatever happens to fly by their faces is what they're going to eat. And two, that with the Chiricahuas, it's a pretty lush environment, and so it just might be that there are a lot of terrestrial insects for them to eat, and since we didn't see necessarily a preference, it really might just be whatever's flying in front of their faces that they're eating. And so for future research, it should examine what is allowing Sclopper scierivi to do slightly better at perennial research than ephemeral ones. Just because we do know that there's probably something going on, and I just might not have been able to detect it, only for the fact that stream drying is not really good for ecosystems in general. And so just further exploration on that is going to be necessary as we continue to see that happen. So a little bit about the Doris Duke Conservation Scholars Program. So I was not out doing this research alone. I actually had a lot of undergraduate students with me. So I had two at a time over the course of 3 years. And so this was a really cool program that allowed undergraduate students to come out and do field research for a summer with a graduate student. And then we would help them analyze the data that they collected. And they actually got to do their own mini projects too. So in addition to helping me, they got to do their own mini projects. I helped them analyze the data from their mini projects, and they created a poster that they got to present at the Ecological Society of America. And then in the following summer, they got to go and work with any NGO, state organization, really, as long as it was dealing with conservation and was in the US, they were able to do it. Some undergrads got to work with wolves, some got to do sea otters, some did like bees and pollinators, birds, whatever they wanted to do. And so that was really cool. So now we're gonna move into lizards at Cabrillo National Monument. And this was probably the one of the things that was most fun about putting this presentation together because I was like, what are some interesting things that maybe I don't know about some of these lizards? So we're going to start with a really cool one, the southern alligator lizard. So it lives along the Pacific coast, and it's found in a bunch of different habitats, but it prefers areas with surface cover because it likes, especially like leaf litter, so like leaves that have fallen down off of trees and things like that or under rocks, but it has this long body and these kind of like shorter legs, because they like to move through the leaf litter or under things to find their food sources, and that kind of like serpentine movement that they do is kind of to mimic snakes. So a lot of times people do think that they're snakes at first, and that's just kind of like to prevent predation. I mixed those two words together, but so they don't get eaten by other things. And like many lizards, they have caudal autonomy, which means that they can drop their tail when necessary. So if they're grabbed by a predator. So I have a couple of demonstration items here, actually. Uh, so I moved back to LA in 2021 after I finished grad school, and that was the first time that I had actually seen an alligator lizards in LA. I was driving, leaving my mom's house, about to get to the main street, and I saw an alligator run across the street, and I parked as quickly as I could, got out the car as quickly as I could, but I did not see the lizard. All I saw left was the tail, so I took the tail for moments like this. So if you think about a lizard's tail, it is in it's more segmented than in like the pictures, but imagine looking at this picture and you see the banding, and imagine that each band is like connects one Lego piece to another Lego piece. So like the lizards have vertebrae in their tails, and at each vertebrae, the lizard can drop the tail if it's grabbed by a predator. So we see how this tail here is really bright and pretty and sparkly. And so this one has a lot less parts. But say a predator grabs this lizard at the tip of its tail. And so the lizard is like, oh, I'm gonna let it go. And then it drops that piece of the tail. Then the lizard has the ability to regrow its tail. However, it is no longer as pretty. It's kind of dull. It has a blunt point at the end versus the sharp point of the original tail. But this lizard is still good to go. It has a lot of its tail left. But say another predator comes along and then it gets grabbed again at the tip of the tail. Well, it can't just drop off this part because that vertebrae it's already been used. It's fused together now. That cartilage is not gonna, it doesn't regrow in that way. This is all one solid piece of cartilage for the tail now. So now it has to go up to the next vertebrae. So it drops its tail here. So now it has to regrow its tail again. And lizards don't want to have to drop their tails because it takes so much energy for them to regrow their tails. But this one, it regrew its tail. But then it drops its tail again. And so now this lizard has lost its whole entire tail. All we have is this base. That magnet was strong. The base here. And so it can regrow, regrow, and regrow until it has more tail. But now we're at the cloaca. If it gets grabbed again, it's getting eaten because it has no more vertebrae to drop its tails at. And so I mentioned the lizard that I saw uh that only left the tail behind. So this is a warning for a part of a dead, well, I don't know if the lizard's probably dead by now because of it was years ago. But this is a non-living specimen of lizard. I just want you to know before I put it on the screen. But this is the tail, and these little white pieces here are actually the vertebrae. And so that you can see how it's kind of like pointed, and so it each point goes into its own little hole in the tail. And so this is the other side of it that each of those vertebrae go into. And so that's how it breaks off. And so at each little segment here, let's see if I can get it to show better. There you go. Each segment where when one layer of scales overlaps the other, that's a vertebrae where it can lose its tail at. So it has a lot of opportunities, and scientists think that the lizards actually have the ability to know, like they can feel where they're being grabbed so that they can only drop as much tail as is necessary, and so essentially like it's up to the discretion of the lizards. But some lizards, if you grab it at the end, it might drop half the tail, which versus another lizard where if you grab it at the end, it might only drop a couple of vertebrae up. It kind of depends on the lizards. Lizards really do have different personalities. And that's a whole nother presentation on lizards that I could do, honestly. But we're running out of time. And so we're gonna move on to the next lizard, which is our legless lizard, which is really cool. That picture is of me, and I finally saw one early. Well, I guess it's not this year anymore. It's last year now, 2024, April 2024. And they are really cool because they spend most of their lives underground, and obviously they don't have legs, but they're still lizards. They have all the same, uh uh things that make other lizards lizards except for the legs. So the same, movable eyelids. They have the shorter body, longer tail. And the interesting thing that makes them different than snakes too is that even though they don't have legs, they don't have the same specialized belly scales that snakes do that allow snakes to grab onto the ground or grab onto trees and allow them to climb up. So like with snakes' specialized scales, even on a smooth surface, the snake can still move, whereas the legless lizards will kind of be floundering because they can't grip anything. They need to have those rougher surfaces because they're not creating their own friction like snakes are. All right, I'm stopping for questions before I go on to the next one. Anything, Sam?
Yes, we actually have three questions in the chat. One of the questions I think we should wait maybe till the very end, because
Okay,
it entails you going backwards and showing a different slide. And that is from someone who wants to learn more about the stable isotope analysis.
gotcha.
But we do have two other questions. How do you determine the sex of lizards?
So it depends on the lizard species. So some lizards are uh they have these post-anal scales, and so essentially this one does not have them, but like they have their post-anal scales, and they're two enlarged ones, one there, and then they will have one there, and they're just bigger than all the other scales. And for these lizards, they have hemipenes, so they have a penis that comes out on either side. And so that's how I know that that's a male, because females will not have those post-anal scales. For other ones, it's more invasive, and you have to go up in there and look, or you have to press on them a little bit to try to get them to express whatever they have going on down there. So it really does depend on the species.
Yeah, did you guys know about hemipenes? It's wild. If you want to go down a very weird rabbit hole, I recommend that you Google lizard or snake hemipenes after this. Okay, and the Shanda is asking, why do they regrow their tails, especially if it takes so much energy? That's a great question, Shanda.
Yeah, because their tails help them with balance. So like when they're falling, and flipping back over with movement and things like that. And there was also a paper actually that recently came out where it's like it was looking at Western fence lizards and regrown tails, and it actually does have like lizard societal consequences if the lizard is like missing its tail or has a regrown tail. So it's better to regrow the tail than to not have a tail at all. All right, so then I'm gonna move on to
Absolutely.
our next
Perfect.
lizard, our Western fence lizard, which I think is probably a fan favorite, also known as bluebellies. Very good at adapting to our urban environments because you find them almost everywhere. And the fun fact that everybody loves is that they have a compound in their blood that actually neutralizes Lyme disease. However, this does not make them a good specimen for getting rid of Lyme in the world, unfortunately. So you can't just like dump Western fence lizards places and be like, eradicate Lyme, because if you like try to dump them in like Minnesota, they're gonna freeze to death. That's not gonna work. But also with more lizards and also more small mammals, they you have more hosts for ticks. And so with more ticks, then you have more chances for Lyme to persist. So yes, they can neutralize the Lyme disease. However, if you have more lizards, you have more ticks, you have more Lyme. So that kind of doesn't really work in that way. And there are scientists who are like looking like how can we pull out the that compound in their blood to like create something in the lab that neutralizes Lyme. They haven't really got quite that far with that just yet. And then we have our Western side blotch lizard, Uta stanisburiana, which are, I think, really cool because of their mating procedures, essentially. So the males have these different throat colors, so they can be blue, orange, or yellow. And the color really correlates with their behavior. So for our blue-throated males, um they're not as aggressive as our orange-throated males, and they're not as timid as our yellow-throated males, but they do try to sit there and defend their territory. But what happens when you're facing one threat? Another threat will come up from behind. Those yellow-throated males will come up from behind and sneak and get at one of the blue-throated females while they're not even looking. And then with the orange-throated ones, they do manage their own territories as well, but then they try to go fight with the blue-throated ones for the territories the blue-throated ones has, and it's just like, is enough not enough for you? Apparently not. And then the yellow-throated ones get to come up again because now the orange-throated one and the blue-throated one is, distracted. And so it's like, oh, okay, I can sneak up in here and get all the females since you guys are fighting with each other. And they kind of just like cycle like that. And I thought that that was like a really interesting thing for lizards to have. Who would have thought that the colors of their throat would dictate how they act when it comes to finding mates? And so this is a really cool lizard that you can find throughout the Western US, and they have also different. Not just throat pattern morphs, but like their actual back patterns and face patterns could look a little bit different too. And they're called a side blotch lizard because they have that very distinctive black blotch right under their armpit. You can kind of sort of see it in this picture as well. And then I got one more lizard that we're going into. I'm trying to stay on time here. So we have Belding's orange-throated whiptail lizard. And so the interesting thing about this one, and we've talked about scales a little bit so far, but these ones have smooth scales versus keeled. So keeled scales are the ones that pop up a little bit. And then you also have lizards that have beaded scales. And so those are, if you've ever seen like a Gila monster, they look like they're literally like have beads on them. They have the little ball scales. And so the different scale types are important for different types of movement. So like how we were talking about our alligator lizard earlier, and then also with this whiptail, those are active predators. So they are actually moving around looking for their prey versus something like a side blotch lizard or a western fence lizard, which kind of like just sit and wait for stuff to come into their face. So if you're moving a lot, you want for any, and especially if you're moving in like sand or leaf litter and stuff like that, you don't want stuff getting caught on your scales. You want to be able to kind of just like glide through whatever it is that you're moving through. Different scale types can help with camouflage. Um, Depending on the lizards or not camouflaging. So like how we were talking about the Gila monster, it is a giant orange and black lizard. It is one of the few venomous lizards in the world and the only one found in the US, and it's saying, "Hey, I'm venomous. You don't want to mess with me. You can see me. That's how you know you don't want to mess with me." So the scales of the lizard can actually like tell you a lot about the type of forager they are, the type of movements that they do, and where it lives on the food web. So I'm gonna stop there again for questions, just because I'm finishing up with the lizards before I go into some of the outreach stuff.
Think the only question we have is that one about the stable isotopes. So let's wait until the very last minute so you can scroll back and show your screen again to that.
All right, cool. So now we're gonna move in from lizards into the outreach stuff. So what the all the lizard studies and the Find That Lizard has allowed me to do, which has been so cool and so fun. So Sam, who is our fearless leader when it comes to our If Then Steam Squad. So if you don't know, the If Then She Can Ambassador program uh essentially put out a call and got 125 women who are leaders in STEM from all different kinds of backgrounds. So both Sam and I are herpetologists into nature and the environment, but we also have there's also like literal rocket scientists, people who do sports medicine, all kinds of things, and bringing them together to essentially for us to work together to help the future, help girls who are interested in STEM have resources that they need to pursue STEM careers. So one of the things that Sam did was she got us together to start working San Diego Comic-Con and putting on community engagement events there. And so we've hosted a number of panels, we've done pop-up museums, we've hosted workshops and things like that. And it has been an amazing time. And Sam, I'm so grateful for you because this is uh, so fun. Um, Other things that I've done, I did a triple AS, so the Association for the Advancement of the Sciences Mass Media Fellowship. So that is for PhD students who are interested in science communication to go and work with a news organization for the summer, and do some reporting, essentially do science journalism. And I'm happy to talk about that experience more if anybody is interested in doing a mass media fellowship, because I think the applications have closed already for this year, but they should open back up in I think in the fall. But I had a really great time. I had a front page news article, which was really cool. I worked for the Las Vegas Review Journal for the summer in 2020, and that was pretty amazing. I've done more outreach on social media. I was one of the original co-organizers of Black Birders Week, which was essentially in 2020 when we were seeing the when we were seeing police brutality more on social media. Not that it was happening more, it was just being shown more. And then there was the incident with Christian Cooper, who was a black birder in New York City, and so he was at Central Park in a protected area, and this lady had her dog off leash. He asked her to put the dog on leash, and she called the cops and told them that he was threatening her, and so he had recorded the whole thing. So, and it wasn't true what she said, but because we know that police brutality is a thing, like she literally put his life at risk. So me and a bunch of other Black scientists, outdoor enthusiasts came together and we were like, hey, why is nobody in our spaces talking about this? Because this affects them because it affects us. And so we wanted to, one, call it out, and then two, celebrate ourselves and each other. So we came up with this and it went super viral, and it inspired a bunch of other Black and X groups and things like that. And so it's pretty cool to have that as a part of your legacy. And then most recently, I was working at the Los Angeles Zoo. I just finished up working there. Um, and I was the coordinator for conservation engagement. So I got to plan a lot of outreach opportunities as well as design or redesign, I should say, the conservation paid internship program for undergraduate students there. And so if there's anyone who knows of an undergraduate who is in Los Angeles and is looking for an internship, I am happy to talk to them about that particular program, which was really cool because I got to have interns come onto zoo grounds for 10 weeks and do restoration research, essentially, which actually did inform some of the work that the zoo will be doing there. I got to design Dungeons and Dragons theme Bio Blitz, and I was really inspired by Sam and the stuff that we did with the Steam Squad for that. And so I was like, hey, I already know that people are interested in science and nature and, Comic Cons and th these different pop culture things. We say we want to build, new communities and new audiences. Here's one way that we can do that. And so that was really awesome to do. And then looking forward, I am planning on continuing to explore my curiosity for nature. I'm working on a Find That Lizard book. I'm doing a lot of pottery and lizard pottery, so sign art, hoping to do more public speaking. And I really kind of want to start to do more on YouTube and things like that. So pretty excited there. And then here is my contact information, and I am going to come back to this slide because I'm going to go back to the slide about the stable isotopes to answer that question. Um, all right, I'm going to
Perfect.
stop here.
And thanks for your patience, Dawn and Marilyn, while we move forward with Erin's presentation. So Dawn and Marilyn would like to know, could you explain what is being presented in the staple isotope slide?
Yeah,
So can you talk a little bit, like explain what each one of these figures means?
absolutely. So these figures, let's see the best way to explain it. It is putting together. So when was out there capturing the insects, I captured a bunch of different insects. And so then essentially I took those samples and I sent them off to the Stable Isotope Lab, and then they gave me back these graphs. And so from those graphs, I had to do uh essentially a statistical analysis to see if there was a difference between the ephemeral and the perennial sites. And so this is the results of that statistical analysis. And so each of these boxes essentially is the representative of the aquatic or the terrestrial insect at the ephemeral sites, and then the ones at the perennial sites, and then comparing the two. And then you can just kind of see that all that those lines in the middle of the boxes are kind of at the same points, and most of the boxes are around the same shapes. And we have um all the what looks kind of like lines going out of the boxes. Part of that is error bars, but then also some of it is probably just some outliers and things like that. And so that's essentially what those individual things are, and that's for each of the different types of insects that we have. So ants, beetles, wasps, aquatic flies, mayflies, caddisflies, moths, terrestrial flies, aquatic beetles, dragonflies, and damselflies.
Got it. So the you're comparing the ephemeral to perennial, and ephemeral are the pink
Yeah,
boxes,
ephemeral to
and
pink
perennial is like that blue
blue.
column, right? And the
Yeah.
lines in the center, that is the median,
Yes.
right? The median result.
Yes.
Um, all right, very good. So yeah, not a lot of difference there, except for this one, maybe down here with terrestrial flies, down here, and maybe some damselflies.
Yeah, and then it still didn't. The statistical
There's,
analysis itself was like, no. It looks
yeah,
like it should be something, and then the stats were like, It's not." So,
yeah, because this number here is so small, and the error
mhm.
bars are so large
Yeah,
too, as well.
they are. They are, unfortunately.
Um, So there is a another question here in the chat before we wrap up. For the stable isotope analysis, did you expect certain insects to be more associated with ephemeral or perennial sites?
Yeah, so I actually was expecting for the dragonflies and damselflies, because looking at previous studies on western fence lizards in California, that's what they saw that they were eating. Um, and so that might also maybe be a behavioral thing because I was also looking at a recent study for western fence lizards that live near coastal areas where they'll actually go down into like the tidal area with tide pools and eat marine invertebrates down there. So maybe the western fence lizards are just more active foragers than uh, the yarrow spiny lizard or the striped plateau lizard. And that's why we're seeing that difference there. So the previous research is why I expected to see something.
Yeah, and it would be interesting to run this research again for a variety of reasons, like not only to get more data at these specific sites with these specific species, but on different species too,
Mhm.
who have very different foraging modes, and to kind of compare and contrast that.
Yeah,
Um, and somebody says tide pooling lizards. I think that's Lauren again. That's so cool. Um, yeah, right. I mean, it's great.
it
You
was. It was
know,
really interesting to read.
and a lot of organisms are opportunistic, right?
Hmm.
Sometimes they're very specific about what they'll go after, and sometimes they're like, "Hey, here's a prey source that no one else is really going after, so I'm gonna get me some of that." You know,
Exactly.
that's great. We are a few minutes over. I just want to open it up to any last minute questions quickly before Erin logs off in the Q&A. And I am going to ask one while y'all, quickly type in your last minute questions. And that is, Erin, we like to leave on a positive note, right? You're a conservationist, just like I am. Is there something that people can do at home? Are there things that you can recommend that people can do in their daily lives or as a hobby to help conserve this beautiful planet we live on?
Absolutely. I would say for sure prioritize native plants. So if you have space for planting, prioritizing our natives is always a super important thing to do. Voting for measures that are climate friendly, environmental friendly, your own spending habits. So, if you can afford to buy something that is not like Shein or Fashion Nova or some like fast fashion, if you can invest in pieces that are ethically made, then definitely doing that, or even like thrift shopping or going to consignment stores and things like that. So not creating more things to be on the earth and you definitely using what we already have, and then just making like small changes wherever you can. Maybe you won't go vegan. I don't think that going vegan is necessarily the answer, but you might be like, I kind of want like fish tonight, so I'm gonna get fish that is, farm raised or that the producer has some kind of like actual sustainability statement or something like that. And then it's also just about doing that research to make sure that it doesn't just say that this fish is sustainable at the grocery store, but you look it up and see, oh, they're actually like, doing what they're supposed to be doing because there's a term called greenwashing where essentially to put it the most simply, companies will lie to make it seem like they're environmentally friendly when they're actually not. So actually just kind of like taking that extra step to make sure that these companies are doing what they say that they're doing, and if they're not, to not continue spending your money with them. So, unfortunately, money makes the world go round. So, your dollar is probably one of the most important things that you can wield against climate change to see a better future. So we have the power, we have the control, and also just having hope and building community and working with each other to going out and volunteering with an organization that you really like working with, donating to an organization that you like working with. we don't have to do it alone. We can't do it alone. We have to do it collectively. And so building community is also something that's really important. And it's just great for your overall health in general, too.
I love that, Arian. What's good for the planet is also good for our health,
Yeah,
and our well-being, right? Community and making sure we have a clean, safe environment. Well,
absolutely.
Erin, I know we're 7 minutes past the hour, so I'm going to let you go. I want
Okay.
to thank you again so much for being here. Let me share my screen one last time here to do a closing slide. And I'd like to thank our sponsors tonight, Cabrillo National Monument Foundation. You can check them out at cnmf.org. And I also wanted to mention that our next Naturally Speaking that's scheduled is on our awesome peregrine falcons, one of my favorite organisms on Earth, with Don Endicott on February 19th. So go ahead and put that in your calendars now. Um, I see there are some good comments in here. Let's see. Hold on one second. Uh, Erin says, thanks so much for having me. Yeah, thank you, Erin. We appreciate you being here. And thanks all of you for coming today. And this is where we say goodbye until next time. Bye, everyone. Have a great night. Thanks for being here.